So the name of my protein is Piezo1, and it's a mechanosensitive ion channel that plays a role in a lot of physiological processes by responding to mechanical stimuli. And this protein can be found in many different cell types, including red blood cells, epithelial cells, endothelial cells, skin cells, and urinary tract cells. And this protein is responsible for mechanosensation. So when it undergoes a gain of function mutation, which is what I'll be talking about today, the protein channel can become overly sensitive and allow a lot of ions in, and this can disrupt the osmotic balance and cause problems with red blood cells, leading to them being more weak and rupturing easily, which is indicative of xerocytosis, which is the disease that's caused by the mutation. So to create my model, I used pipe cleaners, and as you can see, if I hold it this way, it forms a propeller shape. So it's got 3 different regions. It's got your peripheral regions, which are the blades of the propeller, and then you've got your central core, and this is where your ions can travel through your channel. So the primary structure is basically, if you unravel this whole thing and just put it in a straight line, it would be your sequence of amino acids, and there's around 2,500. And these are held together by peptide bonds, which are covalent bonds. And there are different types of amino acids. So you have your nonpolar amino acids like leucine, isoleucine, valine, and methionine. And these are located primarily in your transmembrane region, and they help to form your transmembrane domain, and this is what interacts with your with the membrane of the cell. And then you have your polar amino acids like serine, threonine, and asparagine, and these amino acids, the interactions between them is what helps to hold your secondary and tertiary structures together as a result of hydrogen bonds and ionic bonds that are formed between them. And so our secondary structure is represented by the coils in each of the pipe cleaners, and these are local folding patterns that are caused by hydrogen bonds between the backbone amides and carbonyl groups that are in the amino acids. And the coils are called A helices, and they're present throughout the entire protein. There's also beta pleated sheets, but they're not as common as your A helices. And then the tertiary structure is represented by the different colored pipe cleaners, and as you can see, there are a whole lot of coils and curls, and this arises as a result of the folding of the secondary structure because of side chain interactions. And some of these interactions include hydrophobic interactions between amino acids, hydrogen bonds, ionic bonds, and disulfide bridge formations. And then your quaternary structure would be basically the whole protein, and as you can see, these different colors represent each subunit. And the subunits are held together by hydrophobic interactions, as I previously mentioned, from your amino acids. It's also ionic interactions and hydrogen bonds. And these subunits, like I said earlier, they're going to form your 3 blades called your peripheral regions and a central core, which is where the ions travel through. And so normally, in a normally functioning Piezo protein, you're going to have receptors that respond to pressure, and it's going to cause the protein to open up like this, all 3 ways. And then whenever you reach whatever membrane potential needs to be reached, you're going to close back up like this. However, when there's a mutation, which is represented by this yellow right here, is most of the time it's located in your inner helical bundle towards the center of the central core. And so basically, when there's a mutation there, it's going to cause the protein the channel to stay open for longer than needed. Instead of closing when it's appropriate, it's going to stay open for longer, and it's going to allow a lot more ions than necessary to pass through your channel. And this leads to excessive ion influx, and this is what disturbs your osmotic balance in your red blood cells and causes them to lose water and rupture easily, which is what leads to your xerocytosis. And for the limitations of my model, number 1 is not to scale, obviously. Your protein is going to be extremely small. And so it was kind of hard to make the model to scale. And of course, there's a lot more folding and coils and whatnot, but due to I don't have that many materials, I had to make do with what I had. But of course, there's going to be a lot more coils and a lot more folding and whatnot. And then my second limitation would be that I couldn't show my tertiary structure very well, and so this is going to be over here. Like I said, I didn't have a lot of pipe cleaners, and it's very much floppy, and so there's probably a lot more backbone right here, and there's a lot more folding, and there's also a lot more just structural things that I wasn't able to incorporate into my model because of the material. And then lastly, I can't really show how much the channel opens and closes just because of the material that I used. As you can see at the top, I had to kind of put them all together, but in reality, this top part would also open when the channel will open, but it can't really stand on its own, so I had to connect all of them together. But that's it for my protein, and thank you for watching.